This is a how-to video supporting Pinnacle software. The focus of this video is to explain error-tolerant searching for protein therapeutics. And again, the primary application for this workflow is to support a biopharma application. And the assumptions are that the protein that's under study is well purified or has very little background matrix effects. The known aspect of it is a base protein sequence, as well as the digestive conditions in which the data was acquired. The way that you would set up and perform the study is when you open up Pinnacle, you would go to the preset workflow and select the workflow. And in this case, we're going to perform antibody screening and quantitation. When we click on it, the field to the right-hand side are all of the parameters that we will need to set. And if we start at the top, the section of adding raw files, you can browse and select any specific file that you would like to look at. The next thing that you would do is specify sort of your knowledge base, that is the protein or peptide sequences in which you're going to use as a starting point. And there's two options. You either have the choice of a FASTA file, or you can copy and paste a sequence. And again, what we're looking at today is just a simple protein of fetuin that was been digested using trypsin. And we can utilize either the FASTA file or we could copy and paste this particular sequence. Once you've specified the raw file and the FASTA or the sequence that you're going to use to start with, you would then set up the digestion conditions, and generally a primary cleavage enzyme, in this case trypsin, number of missed cleavage sites to consider. This is for an indexing and initial searching. And then when you go to the antibody screening, this is where you would select your options from which to process the data. In our particular case, we're not going to look for glycopeptides, but just rather the unmodified the peptides that are modified by known or predicted mass shifts. And in this case, we're going to check the box for an error-tolerant search. We can keep static modification, but we are going to get rid of any dynamic modifications. And we're going to allow the error-tolerant search to identify potential modifications. And then once we've done the antibody screening aspects, we would then set up our file reading. That is, what is our expectations for the mass tolerance for extracting the data? Is the MS and MS1 or MS1 and MS2 data acquired on an Orbitrap, that is having high resolution and mass accuracy, and you can set the mass tolerances accordingly. And then you're ready to go. You would just simply hit apply. Again, the key factor for the error-tolerant searching is to eliminate any dynamic modifications, and you're going to allow the error-tolerant search to identify the putative mass shifts, and you can have a static modification. For the fetuin and the raw files that we're going to process, when we hit apply, it will take about 3 minutes to actually process the data. I've already completed the initial search, and we'll just show what those searches would look like. And in this case, we utilize trypsin as our enzyme for digestion. And if we look at the data, the default view would show you in the protein peptide sort of view. We have the table of results on the left-hand side, a chromatographic view, and then we have a mass spectrum shown below. If we expand the table to the left, we get a series of peptides that have been identified based on the sequence and putative mass shifts. And we're going to simply go through a processing step. And in this case, we would start with the unmodified form of a peptide, and we'll just grab this particular peptide. When I click on it, it will change this chromatographic view from all possible peptides that were attributed to the protein sequence to the specific peptide as we've highlighted. And the triangles just simply represent MS2 events or spectra that are attributed to our precursor mass to charge value. So if I click on any one of these or just hover over it, I can see that this is the +2 precursor charge state and was fragmented using CID. Or I have +2 acquired in the HCD cell. So if I click on it, I can either look at the MS2 for this particular peptide, or I can look at the MS1. To look at the MS1, if I just click on it and right mouse click and drag, I can showcase, in this case, 3 individual full scan mass spectrum. And I can expand this out, and I can very quickly see across the entire full scan mass spectrum the +2 charge state, and I can begin to zoom in across 3 different scans to evaluate the isotopic distribution, as well as get an estimate of the mass error. The triangle represents the centered mass or theoretical mass, and you can see where the distribution is, and of course anywhere that I drag my cursor will give me the mass deviation. If we go back to the MS2, again, this is the MS2 for the unmodified form of the peptide. The search results also show us that there's a putative modified form of this peptide, which appears to be an oxidation at the proline. One of the things that we can do is, of course, compare against the unmodified form to see if there is a modification and if the predicted site is correct. In order to do this, we will keep this at a multiplot, and now which shows me the product ion spectrum for the unmodified form, we click on the modified form, and once we click on the modified form, again, we want to match up the product ion spectra, which is the +2 charge state using HCD. So again, if we hover and click, we add in the second spectrum, and you can see that the 2 different spectra are differentiated based on the sequence, and the bottom one has the site of the modification. We have 2 different colored fonts in the product ion spectra, and they represent those ions, product ions, that are the same in both, which are shown in blue. And again, just to zoom in, just to get an idea, this is the B3, and you can see the isotopic fidelity across both spectra. If we zoom back out, we also see the red. And the red represents those fragment ions that are attributed to the sequence, but have a different mass to charge value. And if we look at the high mass Y series, we see that the Y9, Y10, Y11, and Y12 are all offset by roughly 16 Daltons. And again, we utilize the difference to try and localize the site of the modification. This ends the video for the error tolerant search.